In this very last section of Unit 1, we are going to talk about our first organism or first family. So we're going to be talking about the Staphylococcaceae. So again, the organism that we're going to talk about now, the family is the Staphylococcaceae. The genus, which remember, it's just kind of like the organism's first name, Staphylococcus. And then we'll be talking about various species within this genus and family. And if you just want to kind of test yourself, what kind of hemolysis is that? Okay, some of the general characteristics of staph species. So these are going to be characteristics that are true for all of the staphs. So they are catalase positive. We'll get into that a little bit more later. But catalase is the test that differentiates staph species from strep species. They are all gram-positive cocci that are seen mostly in pairs or clusters on the gram stain. They're mesophiles, which means that they prefer body temperature, human body temperature. They are aerobic or facultative anaerobic organisms, except for Staph saccharolyticus. They are non-motile and they are non-spore forming. This flow chart is, I recommend that you kind of familiarize yourself with this. This is a great tool. It's just kind of the big picture, what an overview of the testing that would be done, and based on that testing, just kind of the flow chart to get us to an identification within a staph species. So again, we start out with the gram stain. We know that we've got a gram-positive cocci. Then we would do the catalase test. If it's positive, that tells us that it is a staph. Remember on the preceding PowerPoint, I said in the general characteristics, all staph species are catalase positive. If it was catalase negative, that would be in the family Streptococcaceae, which we'll get into in the next unit. So from them, there are additional tests that you can do, and we'll talk about some more of these in just a minute. Coagulase is one of those that helps us determine Staph aureus from some of the other staph species, and then additional testing. But this flow chart will help you when we're talking about the testing to identify particular organisms. Okay, so particularly talking, we're getting into some of the species now. So Staphylococcus aureus is the only coagulase positive staph species. And again, from the flow chart here, we know they're all gram-positive cocci. If it's catalase positive, we know we're dealing with the staph species. Then coagulase is the next test that we would do to determine, is this organism Staphylococcus aureus or is it one of these other ones? So the first one we're talking about is Staph aureus, and it is the only coagulase positive staph. Colony growth. And Staph aureus is the most clinically significant species of the genus. On the gram stain, we know it's gram-positive cocci, but a lot of time it will appear in clusters. It's beta hemolytic. Very rarely you'll encounter an isolate of Staph aureus that is not beta hemolytic. Your colony size, so that's the actual growth on the plate. They're usually about 2 to 3 millimeters in diameter, convex. They're referred to as buttery looking. They can be white to yellow. And again, they show that beta hemolysis on sheep's blood agar. Some of the testing that we would do to identify Staph aureus, again, catalase positive because it's a staph, coagulase positive because it's Staph aureus. But other testing that we can do to help differentiate, the mannitol salt agar. Remember, that was a plate that we talked about either in the last lecture or the one before that. So it is positive, which means it utilizes the mannitol, so it will change the color of the media. So uninoculated, that mannitol salt agar plate is red. Staph aureus utilizes the mannitol, turns the plate yellow. It's also DNase positive, and DNase is just an enzyme capable of hydrolyzing double-stranded DNA. Some of the normal habitat, so places that you would find it on the human body: nose, throat, skin, vagina, nasopharynx, in about 15% of the population. So those folks would be considered carriers of the organism. So here's your picture of Staphylococcus aureus on the mannitol salt agar plate. And again, MSA is selective and differential. So it is selective for staph species. It's differential. So if you've got an organism that's growing on the plate, most likely it's going to be a staph species. And then it is differential, so we can help differentiate Staph aureus from the other species of staph based on mannitol utilization. So Staph aureus utilizes the mannitol in the agar, turns the agar yellow. Okay, so some different virulence factors associated with Staph aureus. Different enzymes that the organism can produce. Coagulase is one, so this is a clotting enzyme. That's what we're essentially testing for when we're doing the coagulase test. That helps us differentiate coag positive from coag negative staphs. It's not really clear what its exact role in pathogenicity is, but it is considered a virulence marker. So, and there's two different types. Clumping factor, which is also known as coagulase, is a cell-bound associated protein. So on the surface of the cell, bound to the cell surface, it's heat stable. This is what is detected. There's two different methods of performing the coagulase test. So you can do a slide coagulase test or a tube coagulase test. Clumping factor, aka coagulase, is what is detected in the slide coagulase test because it causes bacterial cells to agglutinate in plasma. Free coagulase, also known as staphylocoagulase, is an enzyme secreted extracellularly. It is heat labile, which means it's affected by heat. It's detected in the tube coagulase test. So in the tube coagulase test, there's a clot formed when the bacteria are incubated with plasma. And we'll get into that test a little bit more later. Hyaluronidase is another virulence factor, also referred to as spreading factor. It permits the spread of bacteria by destroying connective tissue. Lipase destroys tissue, facilitates the spread of disease. Catalase is an enzyme present in staph that differentiates it from strep. DNase is an enzyme capable of hydrolyzing or destroying double-stranded DNA. And then beta-lactamase. 95% of clinical isolates are positive for beta-lactamase, which means that those isolates would be able to break down the beta-lactam ring of many penicillins. So penicillin is a class of drug. So if the organism produces this enzyme that makes it able to break down the beta-lactam ring of penicillins, it's going to make this organism resistant to that penicillin class of drugs. Okay, next, some toxins that Staph aureus can produce. Intratoxins. These are heat-stable protein toxins released by the cell into its environment. Food poisoning is the most common presentation of this toxin. Causes violent vomiting and diarrhea as soon as 2 to 8 hours after eating. Can lead to tissue damage. TSST1 or toxic shock syndrome toxin is associated with menstruation-associated toxic shock. There can be systemic effects that can lead to shock and death. Exfoliative toxin, this is also known as epidermolytic toxin, implicated in scalded skin syndrome or Ritter's disease, which would be seen in infants, where the epidermal layer of the skin sloughs off. Bullous impetigo, which means that blisters form. You've got these fluid-filled blisters that can occur in children under 2 years of age. And then hemolysins, we've talked about those previously. So this is alpha and beta hemolysins and leukocidin. So they damage red cells. So we know from the previous lecture that that's what hemolysins do. They break down red cells. but also platelets, white cells, macrophages, which are a type of large white cell that plays a critical role in phagocytosis. And then protein A is a cellular component, has the ability to block phagocytosis and negate the protective effects of IgG. And phagocytosis is the process that occurs in our body when our cells in our body come in contact with a foreign pathogen. There are certain cells that are, their job is to take out those pathogens. So you can think of it kind of like Pac-Man. They go around and gobble up all the pathogens. Okay, some diseases associated with Staph aureus, skin or wound infections, so the patient can have boils, carbuncles, which are large invasive lesions that develop further from the boils, and then acne, food poisoning. Again, this can happen as quickly as between 2 to 8 hours after ingestion, and this is usually associated with mayonnaise-containing salads or meats left out at room temperature. Bacteremia, which is just the presence of bacteria in the blood. Toxic shock syndrome, which we just spoke about. It's rare now, but potentially fatal. Patient can have a sudden onset of fever, chills, vomiting, diarrhea, muscle aches, and a rash. It can progress to hypotension and shock. It's linked to tampon usage, but there have been cases documented in men and in non-menstruating women. Toxic epidermal necrolysis, so this will present with symptoms similar to scalded skin syndrome, but it's usually drug-induced. Some cases are linked to infections and vaccines. The cause is really kind of unknown, but it seems to be linked to some type of hypersensitivity reaction. Then we've got staphylococcal pneumonia. This is pneumonia that's caused by a staph species. It's rare, but it will have a high mortality rate. And then MRSA, which I'm sure all of you guys have heard of, which stands for methicillin-resistant Staph aureus. So this is a more severe form of the infection. And methicillin is, even though the organism is called methicillin-resistant Staph aureus, methicillin is no longer used in the United States, but the name kind of stuck. So we used to use oxacillin resistance to indicate methicillin resistant also, so to indicate an isolate of MRSA. But now cefoxitin is more widely used to detect methicillin resistant. So we started with methicillin, we don't use that one anymore. Then we went to oxacillin to indicate resistance. Now we use cefoxitin. Most oxacillin and cefoxitin resistance is due to a gene called the MECA gene. Gold standard of testing for MRSA is PCR or molecular testing for that gene. If an isolate is found to be MRSA, then the patient gets put on isolation precautions. And vancomycin is now the treatment for MRSA infections. Here's just an image of a baby with scalded skin syndrome. And again, here's the information about MRSA. So cefoxitin is the drug that's used now to detect resistance and to indicate an isolate as MRSA. Okay, so Staph aureus is the only coagulase positive staph species. Now we're going to talk about the other coagulase negative Staphylococcus species. And we're not going to talk about every staph species, but these are just the more commonly seen in the lab, the coagulase negative staphs. So Staphylococcus epidermidis, the colony, so how it appears on the blood agar plate, you'll have smooth white colonies that are rarely beta hemolytic. So our lab ID, it's catalase positive. Remember, that's the test that we use to differentiate staph from strep. It's coagulase negative because Staph aureus is the only of the staph species that is coagulase positive. It's mannitol salt negative. It's sensitive to novobiocin. Normal habitat on the body would be skin, throat, and nose. Some diseases that could be associated with it. Most diseases associated with Staph epidermidis are hospital-acquired. So it can be considered a secondary invader occurring with prosthesis, shunts, heart valves, and catheters, septicemia in immunocompromised patients. So staph epidermidis largely is just a normal skin flora. So most of the time when we recover it from a specimen or an isolate in the lab, it's going to be considered contaminant. But there are cases, especially in those patients who are immunocompromised, where it can be a pathogen. Here's a picture of staph epidermidis on a blood agar plate, and you can see much brighter white colonies than those of Staph aureus, lack of beta hemolysis. Okay, the next coagulase negative staph species that we're going to talk about is Staph saprophyticus. A lot of strains of saprophyticus can take on kind of a yellow color, and it's caused by a pigment called lipochrome. So lab ID, again, catalase positive because it's a staph, coagulase negative because Staph aureus is the only one that is coagulase positive. It's mannitol salt negative. You may see some weak positive reactions. It's resistant to novobiosin. So that's, if we suspect a colony or an organism to be Staph saprophyticus, novobiosin resistance is normally how we would identify that. Diseases that it can cause: UTIs, especially in young females, usually due to sexual activity, rarely found on other mucous membranes. It can be, if it's seen in small numbers, it's still considered significant. And then just some other species of coagulase negative staphs that we won't necessarily go into, but just kind of be familiar with the names and the fact that they are coagulase negative staphs. So Staph intermedius, lugdunensis, and then Hyacus, delphini, and Schelferii are animal pathogens. Okay, here's a picture of Staph saprophyticus on the blood agar plate. It's a very, very kind of bright, bold white, usually is how you'll see it. It really stands out more than the others. And then next we're going to talk about how to differentiate Micrococcus versus Staphylococcus. So Micrococcus is in the family Micrococcaceae. It's a gram-positive cocci that can resemble staph species. It's found in the environment, and it's part of normal flora, but you can confuse it with staph, so you would have to differentiate it from staph. Both are catalase positive, coagulase negative, so you would be differentiating it from your coagulase negative staphs. So the test that we use to do that is the bacitracin disc test. So it's just like a little tiny round piece of filter paper that is impregnated with the drug bacitracin. So we can set up a test using that bacitracin disc to help us differentiate staph from Micrococcus. So staph species will be resistant to bacitracin, whereas Micrococcus will be sensitive. Another test that can be used is called the microdase disc test. So Micrococcus would produce a blue color, and then your staph species would, there would be no color change, so that would be considered negative. Okay, a few little review questions here. Which staph species is coagulase positive? The only one that is coagulase positive is Staph aureus. Which drug do we use to indicate an isolate as MRSA? So it used to be methicillin, then we switched to oxacillin, but now we use cefoxitin. Which test could be used to differentiate an isolate of Staph saprophyticus from other staph species? That's the novobiosin test. The last section of this unit lecture is all lab procedures. So these are the tests that we will be performing in class. So you will need to read over the remainder of these slides and kind of somewhat familiarize yourself with the procedures that we'll be performing in class. Several of these procedures also have related supplemental YouTube videos in Moodle where you can watch the procedure before you come into class. So we've already talked about the gram stain, we've talked about the reagents that we used, we talked about the control organisms, because remember, every test that we perform in the lab has to have a positive and negative control performed in addition to our patient sample, and that's just to ensure that our results are accurate. And then the purpose of that gram stain is, remember, we're categorizing bacteria based on principles of their cell wall. So your gram-positive bacteria are made up of certain things in their cell wall that causes them to hold on to that. Crystal violet, primary stain, gram-negative organisms have a much thinner cell wall that will cause them to hold on to that secondary stain, the safranin. So there's your procedure. Again, read through that, watch the video in Moodle, and then we will be performing that in class. Again, just kind of a refresher on the differences between gram-positive and negative. So this one is purple, so we report it as gram-positive, and it's round. See that round shape? So it's a cocci. So this gram stain would be reported as gram-positive cocci. This one is, if we're looking at this under the microscope, we see pink. So that's gram-negative, and they are rod shaped, so that we report them as either gram-negative rods or gram-negative bacilli. The catalase test, the reagent for that one is just hydrogen peroxide, and we're just trying to determine if the organism contains the enzyme catalase. And remember, this is what we use to differentiate staph species from strep species. So our positive control would be a staph, our negative control would be a strep. And it's just as simple as a drop of hydrogen peroxide and placing a small amount of the bacteria into the peroxide to see if it bubbles. So the positive reaction is bubbling, negative is no bubbles. The coagulase, and remember I said there are two different forms of coagulase test. We can either do the slide or the tube. Actually, really there are more than that, because the one that you guys are going to perform in class is it's called a latex coagulase kit, but coagulase is how we differentiate coagulase positive staphs. The only coagulase positive staph, remember, is Staph aureus, so that has to be our positive control. And then, so we're differentiating it from all of the other staph species, but our negative control, we use Staph epidermidis specifically, and we're trying to determine if that organism produces that enzyme coagulase, which is also known as clumping factor. Remember, that's the one that's present on the cell surface of the organism. You can see the procedure there. The result is agglutination, or it forms these little clumps, versus no agglutination. The coagulase tube test is a little bit different. You use the same reagent, which is rabbit plasma, but it does require an incubation step. And then the tube coagulase test, remember, we're testing for extracellular or free coagulase. So it detects staphylocoagulase, and the formation of the clot is what's considered positive reaction. Controls are the same. It does require an incubation step, though, like I said, and then our results, we're looking for a clot, which indicates Staph aureus. So you can see, I mean, this is a liquid reagent that's been dropped into the tube. It's been incubated for a few hours, and then if you turn it on its side, the negative reaction, the liquid will run up the side of the tube. The positive reaction has formed a clot, so it will not. And again, here's just kind of like the little, the difference between the slide method and the tube method. The one you guys are going to be performing in class is a little bit different from both of these, but it's more similar to the slide test. Mannitol salt auger, again, this is just another way to differentiate Staph aureus from the other coagulase negative staphs, but it's a media. So it's a selective and differential media. We're going to subculture a suspected organism to this plate, and what we're looking for is a color change. So it's selective in that only staph species will grow on the plate. It's differential in that it will differentiate Staph aureus through that color change. The mannitol, the Staph aureus is utilized the mannitol, so it will differentiate based on that mannitol utilization against other coagulase negative staph species that will not. And I don't remember if I just mentioned this or not, but this is one that you will not be performing in class. Novobiocin sensitivity is how we determine Staph saprophyticus from the other coagulase negative staphs. And I mentioned that largely the coagulase negative staphs are considered contaminants. Staph saprophyticus is kind of an exception. It is going to be a pathogen. So if we recover it, most likely it's going to be recovered from urine cultures. We would need to identify and report it out. But as mentioned previously, also, the other coagulase negative staphs could potentially be pathogens. It would just be in immunocompromised folks. So there's the procedure for that. And we probably will not be performing this one in class, but it is a drug disc test. So we have to make a broth from our suspected organism that we swab all over the surface of the plate. So on this side we've swabbed Staph epidermidis. On the right side we've swabbed Staph saprophyticus. We place the drug discs in the middle of that inoculated area. It has to incubate overnight, and then when we come back the next day, we're looking for this zone. So if you see that, it's called a zone of no growth. If you see that zone of no growth around the disc, that means that antibiotic that's in the disc was effective at killing the organism. So the organism is not growing within that zone. Versus on the right-hand side here, Staph saprophyticus is resistant to novobiocin. So it will grow up to the disc, so you do not see that zone of no growth, and that's how we differentiate. Couple methods of miscellaneous testing. So I mentioned that you guys will be doing a latex coagulase test. So this is just a kit or like a box test that uses plasma-coated carrier particles, so such as latex. It can detect clumping factor and protein A in the cell wall of Staph aureus. Some of your coag negative staphs can be positive for the rapid test, but the tube coag test would still be negative. And then MRSA, again, we're doing sensitivity testing against the drug cefoxitin or the MEK-A gene, which is detected by PCR, which is a molecular test. And so here's a picture of your latex staph ID that you're going to perform. This is just a picture of one of the little molecular testing units. So they come with these little cartridges that are made for these instruments, and we have like little media that we have to inoculate and then pour over into the cartridge, and molecular testing happens, and we usually can have a result within an hour to an hour and a half. This is how most of your COVID testing is done now, and there are a lot of other tests that can be performed on this same type of platform. And that is the end of Unit 1 lecture.